Hello, my name is Brooke Roges, and I'm going to be discussing some of the research we have going on at Mayville State University. So I'm going to be discussing our research involving cancer intervention using wheat-bran bioactive compounds. The bioactive compound we will be considering in this study is ferulic acid. It is a hydroxycinnamic acid that can be extracted from a variety of fruits and vegetables, including wheat bran. It has potent antioxidant activity that has shown in previous studies to have an effect on diseases including diabetes, neurodegenerative diseases, cardiovascular disease, and specifically cancer. The objectives of this study are as follows: to successfully culture cancer cells, as well as test ferulic acid concentrations on their cell growth, and to determine DNA fragmentation of these cells. Pancreatic cancer is among the top 5 most deadly cancers. It has a less than 30% survival rate past 1 year, and only 5 to 7% survive past 5 years. The chemotherapy commonly used to treat this cancer can be very detrimental to a person's health, so nutraceuticals and natural products and treatments are a high topic of study. The most vital material we were using in this study were the MIA-PA-CA2 human pancreatic cancer cells that we were graciously gifted to from NDSU. They gave us 2 vials containing 10,000 cells each. We also used a variety of cell media components, including DMEM, fetal bovine serum, horse serum, and streptomycin penicillin. We also used the enzyme trypsin for subculturing purposes. The first method we followed was the ferulic acid extraction. Using the protocol as described here, we were able to successfully extract ferulic acid from wheat bran and sent it to the UND School of Medicine to compare it to the standard to see if we had successfully extracted it and could be used in future studies. The cell culture process was also successful, and we began by thawing the cells and testing them with trypan blue for cell viability, which came back over 50%, so we were able to use these cells. The subculturing process took place every 5 to 7 days, and we were able to get exponential growth from the cells. The final method we followed was the ferulic acid addition. We began by creating less than 1% DMSO stock solutions containing 3 different ferulic acid concentrations. They are listed above. And then we inoculated cells into 96 well microtiter plates at 5.4 times 10 to the 4th cell per well concentration. We incubated these for 3 hours prior to compound addition, and then we added the stock solution at 2 rows each per plate, with 2 rows left for control, and incubated these for 72 hours. We then analyzed for cell death as well as DNA fragmentation. For the ferulic acid extraction, we were very successful in extracting it, and compared to the standard, we were able to use the ferulic acid in our studies. It also resulted in a 287.5002 nanograms per microliter concentration. Cell culture growth was also extremely successful, and from the 10,000 cells of each vial we were given, we cultured them to 8 times the initial amount. And for the ferulic acid addition, we were able to analyze them using dead cell percentage as well as DNA fragmentation. As you can see in the image provided, the higher concentrations resulted in a much higher percentage of dead cells within the samples. These are the results for the DNA fragmentation testing. The red glow is the fluorescence of propydium iodide that shows the dead cells, and the green is the specific DNA fragmentation fluorescence, so the brighter green glow shows more DNA fragmentation, which means the DNA is uncoiling and being fragmented, and you can see in figure C that the 0.10 millimolar ferulic acid concentration had a great effect on the DNA fragmentation. So far we have seen results from adding ferulic acid to cause more cell death and DNA fragmentation within pancreatic cancer cells. We are going to continue to move forward by extracting more ferulic acid from wheat bran to use higher concentrations to analyze their effects. We are also going to be using different bioactive compounds extracted from wheat bran, including alkyl resorcinol, as well as urbinoxylum, a current topic of study in different projects going on our campus. These are my references and acknowledgments. Thank you to ND EPSCoR Track 1C grant for your funding, and thank you for listening.